Hi, my name is Kairavi Desai. I am working under the supervision of Dr. Kharche in the PM3 lab and Dr. Welsh and Dr. Mironova in the Welsh lab for my thesis project, which is titled The Assessment of the Mouse Model to Study Arrhythmia through 3D Anatomy and Ion Channel Biophysics. According to a study by Ruiz de Castro-Villot et al., nearly 40% of the patients examined in a cardiology outpatient clinic and 1 in 4 of the people examined for the first time were diagnosed with a cardiac arrhythmia. But that leads us to the question, what are cardiac arrhythmias? Well, an arrhythmia is a change in the physiological heart rate or rhythm that can result from two major categories of mechanisms: abnormal impulse formation or conduction disturbances. As you can see from this animation, here's the normal heart that's pumping at a normal rhythm. On the other hand, in this heart that has cardiac arrhythmia, the heart rate is abnormal and it's pumping way faster than the normal heart is. Arrhythmias can be classified based on where they originate from and how they change the heart rate. There are two main types of arrhythmias: bradycardia and tachycardia. As seen from the diagram, bradycardia refers to a slow heartbeat, while tachycardia refers to a fast heartbeat. While fast or slow heartbeats can be normal, for instance, after you finish exercising, you may notice that your heart is beating faster than normal. But the changes in heart rate that result from arrhythmia are due to changes in the heart's ability to maintain physiological conditions. An example of a tachycardia originating from the atria is atrial fibrillation, which is caused by chaotic electrical impulses in the atria. These signals result in rapid, uncoordinated recontractions of the atria. Similarly, ventricular fibrillation can be even more severe as it causes the ventricles to quiver rather than pump blood to the rest of the body, which results in the blood supply being cut off to vital organs such as the brain. The mechanisms of arrhythmia, as mentioned before, can be simplified into two general categories, which are abnormal impulse formation or reentrant. For this project, since we are focusing on L-type calcium channel, a dysfunction in the channel can result in early after depolarization, which can then contribute to issues in re-entry. Hence, there's no question that arrhythmias can have severe consequences, including cognitive impairment or dementia, heart failure, sudden cardiac arrest, stroke, or even the worsening of existing arrhythmia. The two main aspects to investigating arrhythmias are considering ion channel dysfunction and examining the structural changes of the heart. To investigate the differences between healthy ion channels and dysfunctional ion channels, the patch clamp technique can be used, which provides electrophysiological data to investigate changes in the properties of ion channels in the cell membrane. On the other hand, to investigate changes in the cardiac anatomy, computerized tomography imaging, also known as CT imaging, or diffusion tensor magnetic resonance imaging, also known as DT-MRI, can be used. CT imaging can produce cross-sectional slices of the heart, which can be digitally stacked to form a three-dimensional image, while DT-MRI allows for the examination of cardiac microstructures and fiber arrangement. Mouse models have long been used for studying cardiac arrhythmia and anatomy and pathology of other diseases. Various types of modifications and interventions are easily used in mice. For example, gain and loss of function modifications and gene editing using CRISPR-Cas9 system can be easily conducted in mice. Furthermore, reporter genes and epitope tags can also be inserted in the mouse genome for advanced phenotyping, which makes gene therapy studies very feasible in mice. Studies in mice also offer the opportunity to study how other organ systems and conditions can influence cardiac function. Arrhythmias can be studied at various levels using mice and tissues obtained from mice such as perfused hearts, multicellular or single-cell preparations, and these studies can provide invaluable insights into arrhythmia mechanisms. On the other hand, large animal models, such as the rabbit shown over here, have heart rates, action potential, shapes and durations, ion channel profiles, and intracellular calcium handling systems with dynamics that are more like those seen in humans. Therefore, large animals play a key role in preclinical studies, but it remains very difficult and expensive to perform gene targeting in such models. Hence, the model system for arrhythmias has generally been mice, as they are a more feasible and inexpensive model system. While mice are widely used, their suitability for measuring arrhythmia has not been thoroughly studied. Investigating the utility of the mouse model for studying human arrhythmia will assess its suitability as a model system to recapitulate human disease, specifically arrhythmia. This brings us to our objective and hypothesis. So the objective of my thesis project is to investigate if the mouse model will facilitate the investigation of the human arrhythmia associated with ion channel dysfunction and concomitant structural changes. We hypothesize that changes in the L-type calcium channel in steady states and time constants lead to early after depolarization, and anatomical defects can increase the chance of re-entry formation using the mouse model. The patch clamp technique is an electrophysiological method that allows for the recording of whole cell or single channel currents across cell membranes or other types of biological membranes via ion channels or active transporters. There are 4 types of patch clamp methods: whole cell, inside out, outside out, and cell attached. For our experiment, we will be using the whole cell patch clamp technique, which involves damaging the section of the membrane that is connected to the gigacell, and by applying negative pressure and using a solution identical to the intracellular solution, the membrane potential of the cell can be controlled. This method is primarily used to study the biophysical properties of plasma membrane ionic channels in cardiomyocytes and neurons, making it the perfect technique for us to use. The basic process includes 3 main steps: extraction of the cells from the animal, digestion of the cells to isolate them, and visualization using an apparatus similar to the one shown here. To investigate ion channel dysfunctioning, we will be using the patch clamp method, and to first learn how the technique works, we will be using smooth muscle cells from the cerebral artery, medial cerebral artery, and posterior cerebral artery of the mouse brain. With these channels, we will be isolating for the L-type calcium channel. Upon researching methodologies used for the extraction and digestion of cardiac myocytes in previous literature, we will perform whole cell patch clamp on the heart cells, again isolating for the L-type calcium channel following some of their procedures. The cells will be in one of 2 conditions: healthy control or experimental arrhythmic. The cells will be extracted from the atria, after which they will be mechanically and enzymatically digested. The calcium channel was measured using the following voltage clamp protocol. The membrane potential of myocytes was stepped for 250 milliseconds between -60 and +60 millivolts in 10 millivolt increments immediately after a 500 millisecond pre-pulse to -45 millivolts to inactivate the sodium channel current. The holding potential will be at -70 millivolts, as that was what worked best in previous literature. High-resolution imaging data will be obtained from open-source repositories, including the tomographic dataset of a whole mouse heart via phase contrast micro-CT, which would look similar to this, and DT MRI, which would look similar to this. In total, an atlas consisting of 3 to 5 mouse hearts will be generated. Using a provided MATLAB imaging processing program from the PM3 lab, I will reconstruct images into 3D cardiac anatomies and generate fiber architecture that represents the cardiomyocyte orientation in the heart. The second MATLAB program will use the collected ion channel data from the patch clamp technique to process it and extract the changes in biophysical parameters such as voltage and current due to arrhythmia. From generating an atlas consisting of 3 to 5 mouse hearts, I anticipate that the assessment of structural changes in mouse hearts, such as fibrosis, may require further development of imaging protocol. Furthermore, arrhythmia is expected to prolong inactivation time constant of L-type calcium ion channel, which will then lead to early depolarization, which cascades to delayed depolarization and ultimately results in worsening of arrhythmia. It is anticipated that the mouse model will be useful for studying arrhythmias caused by abnormal impulse formations, but may have limitations in investigating arrhythmia related to persistent re-injuries due to a larger wavelength. Hence, the significance of this project is that evaluating the mouse model for cardiac arrhythmia will contribute to the understanding of its utility for studying human disease. Furthermore, it can help to further expand the current physiological knowledge of the mouse model as well as cardiac arrhythmias and contribute to the scientific community. I would just like to say thank you to the following resources that helped me prepare the material for the following presentation. Thank you for listening to my presentation. I would also like to thank the Loss and Health Research Institute, Robarts Research Institute, the Western University, and specifically the Schulich School of Medicine and Dentistry for providing me with this opportunity to engage in a research project. If you have any questions, please feel free to let me know in the comment section below. Thank you.